This is part 2 of our investigation of the nervous system and control of movement, chapter 7 in our exercise physiology text. In the last section, we learned that action potentials are moving regions of changing membrane potential triggered by initial depolarization by a neurotransmitter. So a neurotransmitter binds to its receptor channel. Sodium ions may enter the cell and depolarize it, making it less negative, and that triggers the opening of electrically gated sodium channels. Those sodium channels are placed all along the axon of a neuron and all along a muscle cell membrane, so that once one opens, sodium ions rush in and depolarize the membrane, and the neighboring sodium ion then opens and depolarizes the region, and so forth. So this top picture represents a part of an axon at rest. There's a net negative charge inside, which I've drawn in with little blue minus signs, and a net positive charge on the outside. That's the negative resting membrane potential. And there's a little voltmeter up here showing us the voltage there, minus 80 millivolts. It's supposed to be what's reading on this graph, although the graph happens to say minus 70 millivolts. But just imagine we're monitoring one spot along the axon with this voltmeter, and we're recording its findings here in graphical form. So then we have this initial depolarization, which will trigger the opening of an electrically gated or switched sodium channel, which is what the black arrow represents, and a sodium ion can rush in, depolarizing that region. Right? If it was negative and now positive charges are coming in, it's depolarized. That's what triggers the next membrane sodium channel, electrogenic sodium channel. More sodium ions come in and depolarize, and so forth, and it just spreads like a wave down the axon. And so as this wave of depolarization passes by our voltmeter, we see depolarization going from minus 70 millivolts, minus 60, minus 50, 0, up as high as plus 30 millivolts as more and more positive charges enter the membrane. And then those sodium channels inactivate, stop letting sodiums in, and around that time, a slower responding set of potassium channels that responds to this depolarization start to open. And when potassium channels open, potassium ions diffuse out. So if you'll remember, there's a high concentration of potassium inside the cell, so when we open up a channel, it diffuses out where there's low potassium concentration. So anyway, letting positive charges out leaves behind a net negative charge again, and we're going back to our resting membrane potential. So repolarization means going from that plus 30 millivolts back down to minus 70 and rest. So I've tried to show the membrane as this wave goes by, going back to a negative interior where it had been depolarized and showing a positive interior. So if that helps you at all, this is another way of illustrating a moving sort of front of depolarization here, and then repolarization as an action potential sweeps down the membrane of this axon. Same thing would be happening on the skeletal muscle cell membrane. We'll discover that more in our next chapter. So neurons are strung together in circuits, with each neuron having some axon terminals, the ends of their axons, associated with the dendrites of another neuron in a little gap between them called the synapse. So an axon terminal can release neurotransmitter that will impact the postsynaptic membrane at a synapse. The neurotransmitter is released from the axon terminal when it depolarizes. And then that binds to receptors, as we've said, on the postsynaptic membrane, and those receptors are channels which open, allowing ions to cross the membrane. That's how one neuron can signal to the next neuron. So an impulse travels down one neuron's axon, neurotransmitter jumps onto the next neuron, the postsynaptic neuron, and if it depolarizes sufficiently, an action potential will form on its axon, and so forth. So here's a little diagram of a postsynaptic neuron, right? It's got some dendrites, its antennas, and here's a presynaptic neuron, which may have an impulse traveling down its axon, and when that impulse or action potential reaches the terminals, neurotransmitter will be released from the terminals into the synaptic cleft, or the synapse, and the neurotransmitter will bind to its receptors, which are ion channels, and ions may then cross the membrane. So that's the mechanism of signaling between neurons in a circuit in the nervous system. Now it turns out that things aren't quite as simple as we've said. Just to reiterate what we've already learned, when neurotransmitters bind to receptor channels that let sodiums into the cell, they depolarize the membrane. That's called excitation, because as the membrane depolarizes more and more, once the axon reaches threshold, the amount of depolarization needed to open an electrically gated channel and form an action potential, then it's excited. So that's called an excitatory postsynaptic potential, or charge, that depolarization. The more it depolarizes, the more likely an action potential will take place. Well, if we look back at this picture, there's many axon terminals here, and each one can cause sodium ions to enter the membrane, and they may diffuse along these dendrites and along the membrane of the cell body to the axon. And the question is, will enough depolarization occur during this process to reach threshold at the axon? Because that's where the electrically gated channels begin, and then they are studded along the membrane for the rest of its length. I guess the axon's over here. I see some myelin sheath. So summation means all of the different events impinging on the dendrites of a neuron and determining whether or not the axon will be sufficiently depolarized to trigger an action potential. So summation is that additive effect of the different events. However, some neurotransmitters bind to receptor channels that are specific for potassium instead of sodium and potassium. In this case, positively charged ions are going to diffuse out, and if that happens, the membrane becomes more negatively charged. Hyperpolarized. When a membrane becomes hyperpolarized, it means it's that much less likely that the axon will reach threshold and have an action potential. So we call it an inhibitory event. So inhibitory neurotransmitters bind to potassium channels and hyperpolarize the membrane. Also, receptor channels that are specific for chloride ions. Those are negatively charged ions that diffuse into the membrane and also hyperpolarize it. So either of those two types of receptor channels will inhibit the postsynaptic membrane. So a given neuron in the central nervous system might have thousands and thousands of axon terminals associated with its dendrites. And the question is, at any given instant, if we sum together all the excitatory postsynaptic potentials or depolarizations from all those terminals and all of the possibly existing inhibitory postsynaptic potentials, hyperpolarizations, what will be the net effect at the axon? That's called summation. Temporal summation. What if a whole bunch of bursts of a neurotransmitter come out of an axon terminal that is excitatory? Every time another burst of neurotransmitter comes out, more receptor channels are going to open, more sodium is going to enter, and more depolarization will occur. That's called temporal summation because it's rapidly succeeding events at the same terminal. They could be inhibitory events, still called temporal summation. Any one axon terminal is only going to release one neurotransmitter, so we're either going to have a whole rapid series of excitatory postsynaptic potentials produced or inhibitory potentials from one particular terminal. Spatial summation refers to the additive effect of multiple different terminals releasing neurotransmitter at the same time in different spots on the dendrites of a postsynaptic neuron, and the additive effect of, like, say, multiple depolarization events, or a depolarization event and a hyperpolarization event, and so forth. So when we sum up all those thousands of different events taking place at the same time, will the axon reach threshold and trigger an action potential? Will it reach, say, minus 50 or 60 millivolts, which is the point where the electrically gated channels open, and then we have that all or none thing happening, and action potential sweeps down the axon. So that's just a little sort of preliminary to think about the complexity of the nervous system. Billions of neurons, thousands of synapses at every neuron on its dendrites in the cell body, and all that summation happening in every neuron in your brain all the time. to determine whether or not action potentials will carry information from one part of a circuit to the next. All right, let's back out a little bit more now and look at the whole nervous system schematically and conceptually. We said at the very beginning that you could think of the nervous system as three simple parts: sensory inputs, sensory neurons that receive information and bring it to the central nervous system. And here's a list of some possibilities: some somatic sensations, things you're consciously aware of, special senses like sight and smell and hearing, and also joint and muscle activity, proprioceptors, produce that information. And then finally, or in the middle, an integration center, billions of neurons that collect all that sensory information and do a lot of processing to determine what to make of it. And then motor outputs, motor neurons carrying outgoing impulses that then will command muscles and glands and make changes in your body. So specifically, proprioceptors, what are they? They are a type of sensory receptor or a class of sensory receptors that provide information to the central nervous system about joint position, joint movement, both direction and speed, muscle length, and muscle tension, tension on the tendons. All of those pieces of information are being reported for all of your joints and muscles all of the time by proprioceptors. The planning centers for moving your body in a way that you desire to accomplish some task need to know what's already happening in your body in order to plan the appropriate pattern of muscle contractions to change and do the thing that you now want to do. So we need to have some data clearinghouse for all this proprioceptor information that we can consult in order to know how to move the body to reach some goal. So we have all these proprioceptors. Kinesthesia just refers to your conscious perception of the position and movement of your limbs, your joints. So what kind of receptors, what kind of nerve endings provide this proprioceptive information? There's free nerve endings, which are just pointy, sharp little dendrites, sort of like that have been drawing in a lot of our cartoons. This little sharp thing might be the way a free nerve ending would look on the dendrite of a sensory neuron. The cell body wouldn't be right there, but anyway. So free nerve endings are one type of proprioceptors. They're all in the joint capsules and around joints, and they allow us to determine the position and movement of the joints. Golgi type receptors are found in tendons. They're called Golgi tendon organs many times, and they detect the tension on the tendon. The more a muscle contracts, the more tension is placed on the tendon, and that's what the Golgi tendon organs respond to and report on. Pacinian corpuscles are a type of collagen fiber encapsulated dendrites. So instead of just a sharp little dendrite sticking out into space, it's a ball of layers of collagen fibers on it. Ruffini's endings are another type of encapsulated nerve ending that could be found in joints and joint capsules and around joints, so that we can recognize when a joint is moving and which way it's moving and so forth. So all these receptors can determine joint position and movement. Muscle proprioceptors. Well, muscles have a type of proprioceptor built into the muscle tissue that can sense the changes in muscle length. So ensconced in and among all the contracting muscles, muscle cells or fibers that create tension are some specialized ones that produce the proprioception information. So that type of receptor is called a muscle spindle. There's also Golgi tendon organs, again in the organ or in the tendons, that detect the tension on a muscle. So we have muscle spindles detecting muscle length or changes in muscle length in the Golgi tendon organ, reporting force of contraction. So what exactly is a muscle spindle? A muscle spindle is a little package of some specialized muscle cells called intrafusal fibers. The fibers that cause the tension, the major fibers that make muscles do what they do and move your skeleton, are called extrafusal fibers. The muscle spindles are made up of intrafusal fibers, all packaged up with a sensory neuron sort of woven around the fibers, so that when the fibers either stretch or shorten, the sensory neuron detects the changes in length and reports that to the brain. It just so happens that there's a type of motor neuron that goes specifically just to the muscle spindle, so that the spindle, if you can imagine the muscle spindle is some muscle fibers in a little package, and as the muscle shortens, they're going to kind of bunch up and get slack. And if that happens, they're not going to report anymore further changes or shortening of the muscle. So gamma motor neurons just kind of tighten them back up again so that we can detect further shortening of the muscle with this muscle spindle. The main motor neuron that controls muscle contraction is called an alpha motor neuron. So that's just a specialized part of the muscle spindle to keep it working over a wider range of muscle lengths. But the point is, it detects changes in muscle length. It's actually interesting because it's the receptor that we're activating when we do a muscle stretch reflex. The knee jerk reflex is just one example of a muscle stretch reflex. Here's a little cartoon depicting muscle stretch reflex in action. So here's a muscle, part of a muscle, and you see some extrafusal fibers here, the ones that do the work. We see a little package of intrafusal fibers. This is a muscle spindle, a type of proprioceptor. And wrapped around those intrafusal fibers are sensory neuron dendrites, which when those intrafusal fibers change length will report to the spinal cord. You see this sensory neuron carrying impulses to the spinal cord. When that happens, the sensory neuron synapses with an alpha motor neuron, causing muscle contraction. So, muscle spindles, when stretched, cause reflex muscle contraction. It's thought to be a postural effect to automate maintenance of muscle tone to maintain body position, body posture. So that if a muscle starts to shorten, or starts to stretch, indicating that you're kind of collapsing a little bit, your posture is wilting, the muscle will automatically collapse by a reflex action, not something that you're aware of, but by a reflex that just automatically causes a little bit of muscle contraction to put back some extra muscle tone to shore up your body position. So anyway, that's a muscle spindle that when stretched produces reflex contraction. And it's a way of testing the sort of sensitivity of the nervous system. Your doctor takes the little rubber handle and hammer and smacks your patellar ligament right here. What's happening is it's tugging on this quadriceps muscle, this rectus femoris muscle, and stretching the spindles, and the spindles cause reflex contraction, and that causes you to kick your foot out. That's so-called knee-jerk reflex. Golgi tendon organs are found in the tendons attached to the muscle, and they detect increasing tension. As the tendon slightly stretches and kind of tightens up during muscle contraction, the sensory neurons that are woven into a Golgi tendon organ start to fire, and they cause reflex inhibition, reflex relaxation of the muscle. So as the muscle contracts, force development occurs. The Golgi tendon organ starts to report and causes reflex relaxation. It's like a safety factor, a limiter of muscle tension. The more tension you start to put on a tendon, the more this reflex starts to interfere with that and limit it. You can't put more than a certain amount of tension before the Golgi tendon organ and Golgi tendon organ reflex blocks you and doesn't let any more tension be placed on that tendon or by that muscle. Interestingly, if you decide to do a strength training exercise program, you can train yourself to kind of ignore the Golgi tendon reflex more than you used to, so that more tension can occur on a tendon before the Golgi tendon reflex starts to really inhibit further tension development. And that's where your initial strength gains come from in a strength exercise training program, is by defeating this Golgi tendon reflex just a little bit. And then eventually, months later, you start developing muscle hypertrophy, and you actually start to get buff, and you gain more muscle strength by just virtue of the size of the muscle cells. So this is just a little schematic of that happening. Here's a tendon of a hamstring muscle here, and there's a Golgi tendon organ in there. And you can see the sensory neuron all woven into the collagen fibers. And as the tension increases, this neuron starts to fire, and in this case, it's going to inhibit the alpha motor neuron. There's a little interneuron here that releases an inhibitory neurotransmitter. IPSPs, inhibitory postsynaptic potentials, are going to be formed on this alpha motor neuron and block further contraction, further increases in tension on this hamstring muscle. So now you see an example of where EPSPs are occurring at this synapse, activating this little interneuron. It's an interneuron because it's between a sensory neuron and a motor neuron. But IPSPs are being induced on this alpha motor neuron, and it's being inhibited. It's not going to produce any additional action potential frequency. All right, so that's just a sort of an intro or primer on proprioceptors and their function in controlling the musculoskeletal system. There are other types of receptors important in exercise, thinking about exercise in the nervous system. There are chemoreceptors in muscles that detect pH, hydrogen ions. The concentration of hydrogen ions is what we call pH. The more of them there are, the more acidic we call the pH. Carbon dioxide accumulation. Potassium ions building up from so many action potentials and repolarizing of membranes, and potassium ions leaking out of the cells during that repolarization phase. Anyway, all the chemoreceptors report to the brain on the degree of metabolism that must be taking place to produce all these waste products of metabolism and exercise activity. So this is what helps control our cardiovascular system. How much extra blood flow is going to be needed? How much extra ventilation, air are we going to have to pump in and out of the lungs to get enough oxygen to that increasing blood flow through the lungs and then to the muscles? But it also probably limits the exercise bout that we undergo so that we don't overdo. Let's talk about that again a little bit more in a second. Somatic motor function refers to voluntary motor function, and that's talking about the skeletal muscles. Anything to do with the skeletal muscles, that's the somatic motor division. So we have motor neurons in the motor cortex of the brain, which synapse with motor neurons in the spinal cord, and those motor neurons in the spinal cord then emanate out to the periphery and form neuromuscular junctions. They're called the skeletal muscles, and activate those muscles to contract. So that's how we turn on a muscle. We want it to contract, we just send impulses down a motor neuron to that muscle. Neurotransmitters are going to come out. A motor unit, it turns out that each motor neuron is wired or innervates not just one muscle cell, but a group of muscle cells. So the group of muscle cells innervated by a particular motor neuron is called a motor unit. Every time that motor neuron is having impulses, that same set of muscle cells will contract, none other. So how do we regulate tension during muscle contractions? It has to do with activation of motor units. Right? You know that you can do a gentle contraction with a muscle or a vigorous contraction with the same muscle. How do we do that? It's done by motor unit recruitment. We start out activating a few small motor units. In other words, motor neurons is just a few muscle cells that they innervate. And if that tension isn't sufficient, right, we have already set a goal in mind for something that we want to do with our body. If that's not a sufficient amount of tension to lift the load or whatever, we're going to activate more motor neurons and the cells that they innervate. And as more and more muscle cells are contracting, more tension or force is going to be generated, and eventually we'll reach a sufficient force to meet our needs. So that's called motor unit recruitment. We just keep on activating more motor neurons and their muscle cells, in other words, motor units, until we get enough tension. Well, here's another really cool thing about recruitment. We just learned about recruitment, now we're already sort of building on that. But when you start out activating a muscle contraction, you start out recruiting small motor units. Smaller motor neurons with small numbers of muscle cells attached to them. If that's not sufficient tension, it doesn't produce sufficient tension, when we recruit more motor units, they are larger motor units. You don't just keep doing the same thing incrementally over and over. We start recruiting larger and larger motor units, so the tension just wraps up exponentially and we really start to get somewhere. So More and more recruitment leads to larger and larger motor units being activated. Here's a cartoon illustration of recruitment happening. Let's say we activate during the initiation of a muscle contraction this purple motor neuron. And whenever the purple motor neuron has impulses, you can see all the little axon terminals are going to release neurotransmitter and activate contraction in these two muscle cells that we see attached. That's one motor unit, one neuron in its two cells. If that isn't sufficient tension, we're going to add simultaneously, send impulses down the second motor neuron, which has at its disposal 3 muscle cells that it innervates. And so now we have 5 muscle cells contracting and an increase in tension. But as we were saying, we don't just keep on adding motor units with 2 cells per motor neuron. We start ramping things up. More muscle cells per motor neuron every time. So not only does the number of muscle cells per motor neuron rise during recruitment, it also varies greatly among different muscles. Tiny muscles that have a high degree of finesse, such as muscles in your hands or especially muscles in your eyes, have very small motor units, a small innervation ratio, that's called. That means that for every motor neuron, there's a small number of muscle cells. Say in the case of your muscles that move your eyeballs, 23 muscle cells per motor neuron, it's been discovered. What about in your thigh muscles, for example? There might be 1,000 muscle cells for every motor neuron, because we know we want big time tension right from the get-go, and then we keep on and we ramp up from there, maybe 5,000 muscle cells per motor neuron as we recruit. So that's called the innervation ratio. So next time, we'll carry the story a little farther as we discuss more about the nervous system, more aspects of nervous system function. How does the nervous system, how do we control with our brain which muscles are going to contract and when? How do we get that information? How do we plan that? That's going to be kind of the subject of next time. So join me for part 3 of Chapter 7, the nervous system structure.